butter sandwich kind of thing. And then there are disulfide bonds that kind of function like the toothpick in the sandwich kind of thing. They really anchor the two polypeptide chains inseparably with each other. And so we went ahead and talked about the so the details of this kind of bread and butter sandwich structure here. I'll remind you what beta sheet structures look like here and so forth, your biochemistry and so forth. I'm not going to ask you to reproduce this structure on the first test or anything like that. I just want you to remember that the immunoglobulin fold, its principal component of it are these beta sheet structures. Like if your multiple choices, one of the choices was alpha helical structures, and the choice was beta sheet structures, go for the beta sheet choice, okay? That kind of thing. I also want you to remember it's a very stable way of, once evolution discovered this folded protein folded structure, it turns out the mean streets of the blood circulation, the extracellular world, these proteins with this fold used to be an old Timex watch ad about takes a lick and then keeps on taking kind of things, okay? It gets beat up, but gets up every time it gets beaten up. Okay. Now then, so now we continue with the new material. And I'll pause a moment here to say that as Thomas did last time, we're going to have an exam review this evening, this afternoon, for the exam one, which will be this coming Friday. And I'm going to go through the lecture notes that we went through most of this time since we've been doing this, and so we'll go through pages, don't worry about this, we're shooting on this, etc. If some of you neglected to bring this handout with you, this evening's lecture, I brought a few with me, just for those of you who didn't quite realize how important it would be to have this with you this afternoon for the exam review. Okay. I hope most of you did remember to bring it for the exam review, because I've only got a handful of folks that are going to be a shark fest in bloody water here, I guess, when I put them up on the table. Okay. But before we do the start of the exam review, which we'll do at the end, I'm going to finish up this set of lecture notes this afternoon. So all this stuff on, come on, do this to me. I told you to autofocus. I keep telling you to autofocus. Why don't you autofocus? How about I told you that? Yeah? It's a good machine. No, I didn't say that. It's really good. Focus. That up. The wrong button. And now the machine will go into a brain freeze and not respond to anything I do. Let's try this again. There you go. Stay with me. Stay with me. You can do it. When in doubt, reboot. Okay. So we're going to finish all this stuff this afternoon, and then whatever time is left, we'll do it on spend on exam review. I hope we'll have 40, 45 minutes left for exam review. But I'm going to do the review. If some of you can't stay a little bit late, I guess tough noogies, I'm just going to talk as long as I takes, and you're welcome to stay on and listen. If you need to get out and get your 4 o'clock cup of coffee, and that takes priority, well, God be with you. But anyway, we'll do what we're going to do here. So, let's get back to the science. So, now this is a representation. So here's the antibody protein. Here's the heavy chain, constant, variable. Okay? The variable domain is a kind of a twisted sister beta sheet structure. It's a little weird, but it's basically recognizably 2 beta sheets on top of each other. But it's a kind of a weird looking beta sheet structure. This part, the variable domain, differs. Each B cell, which is made of genes towards an immunoglobulin M, which is expressing that in cell surface, and an IgM protein, which is a physical membrane protein, has a constant region tail, and then of course the variable segments on here on the arms. Each B cell has made a unique gene that makes a unique IgM protein, and what makes that IgM protein unique is the amino acid sequences out here, the variable part of the protein. And it turns out, as I said, that each antibody will differ from each other in the variable part here, but there's only about a 1 in 50 chain, about a 1 in 50 difference. There are 50 different possibilities for the yellow part of the variable domain, as we'll be discussing. But what really makes each antibody protein and the gene that encodes it unique is the hypervariable part out here at the tips of the variable domains here. So we have a variable which includes a hypervariable region right here. Okay? This is quite variable between different antibodies, but it's not. There are only 50 different ways you can make the yellow part. And each antibody protein will have one of those 50 different ways. Okay? But the hypervariable part is truly unique. Okay? And we're going to talk about how that happens later. So the structure of the V domain that we're showing you here it is right here. It's a kind of a weird looking beta sheet here. And it's all one continuous. Here's the amino terminal end of the protein, and it takes this little way, and then it comes out here as the carboxyl terminal end, and going on into the constant domain down here. There are 3 little loops here, which are right out here at the tip of this box. Here they are. These are the so-called hypervariable loops, or the hypervariable regions of the V domain, and they're color-coded red. So there are 2 different terminologies that you see in the literature. One is to call these hypervariable region 1, hypervariable region 2, hypervariable region 3. An alternative, equally widely used terminology is complementarity determining region. Complementarity determining region, CDR abbreviation. There's a CDR, the HV1, we could also call that the CDR1, CDR2, and CDR3. Okay? If you look at the variability of a bunch of sequenced proteins here, you see all kinds of variability here in the yellow part, and here and here. But then when we come to these loops here, lots of variability here. So these are the hypervariable regions here, and these are the variability in the amino acid sequences that we would measure if we were to sequence a bunch of these proteins. Okay, and then if we were to look at the linear polypeptide sequence, the linear amino acid sequence here, starting from the amino terminal end, wending our way through and coming out of the carboxy terminal end, just laying that out like a string of spaghetti. Here's the amino terminal variable domain, okay, and these regions right here, which are shown in yellow of this structural crystallographic structure, these are called framework regions. Okay, so we have framework region number 1 beginning here and in yellow, and that will wend its way around here, and it will then come out here to form hypervariable region 1 here. Okay? Well, here it is right here. Hypervariable region 1, also known as CDR1. Okay? And then more yellow framework region. And then hypervariable regions here. Framework region number 3, hypervariable framework region number 4. Here again, the carboxy terminal end. Okay? And of course, this combined red loops region right here, this is what's going to bind to the surface of a protein, typically, for example, here. So it'll be, the protein antigen may have little protruding stuff and little indented stuff, and it may have positively charged amino acids and negatively charged amino acids. And this region for the antibody, if the protein has a cavity, this part right here will poke into the cavity, okay? And if at the bottom of the cavity of that protein there's some positively charged amino acids in the antigenic protein, the antibody will have some negatively charged amino acids at the tip of the part that's poking into that cavity, which has a positive base of the well there. That kind of lock and key type of thing is what happens. And we might have a region on the surface of the protein deep in that's kind of hydrophobic, and we might, so for some proteins, this red part here will be a real long loop like that, and it can reach deep into the interior of the protein sometimes. Okay. Anyway, so all this stuff here is, this in red is the epitope binding region. So on the surface of the protein, there's an amino acid sequence made up of all different, the folded structure of the protein. There's some little set of amino acids with a certain charge and hydrophobicity and structural features to it that the antibody is binding to. That part of the protein that the antibody is binding to is called the epitope. I'm getting a little bit ahead of myself. I just want to say that. Okay, so now here is this slide where we talk about epitopes here. Okay, so the part of an antigenic protein to which an antibody binds. Now, the antigen doesn't have to be a protein. It often usually is, but it can be a nucleic acid. It could be a polysaccharide. But the part of the macromolecule antigen here that the antibody binds to, that little string of amino acids is the epitope. So here is a picture. I think this is a poliovirus here. Yes, this is a poliovirus, okay? So here's a poliovirus particle, and it's made up of a kind of polyhedral shell, and it takes 3 different kinds of proteins here, coded, or let's see here, in yellow, blue, and red or pink are the 3 different kinds of proteins that make up this polyhedral shell. And here is the blue proteins here. If you just look at one of the isolated blue proteins, this part of one of the blue proteins, this is bioprotein number 1 here. Here it is isolated, here it is as part of the shell. There's some amino acid sequences here. They're not, in the linear sequence of the protein, we might have amino acid 27, 28, 29 sequentially. Well, sometimes that's the epitope, but more often it's, so all of this in white is one epitope. The amino acids sequence, the amino acids here that are being recognized by the antibody that's coming in and binding right here, they're widely separated from each other if you were to have the extended polypeptide chain just linearly displayed. That linear string of spaghetti, you can curl it all up, they form a little patch where you have amino acids, some of them a few sequential amino acids. Together they're in the folded protein, not too far apart from each other, and this is what the antibody variable domain is going to bind here. Okay. The variable domain made up of the heavy chain hypervariable and the light chain hypervariable things. They're binding. This is now an epitope here. Now actually, I may plural here. This might be one epitope, and other people have an antibody that epitope is commonly recognized by infected people. This might be a second epitope right here. I was thinking to myself as I was talking, that's kind of far apart from each other to me. This is more like the size of an epitope right here. And this is on the size of an epitope. And is it me who's making that popping noise? Oh, God. My God. Stop it. It's fine. It's fine if I leave it on the table, but if I try to put it attached to me, it's going to freak out. Okay. Maybe I'll lower the volume just a little bit. Talking. Okay. Let's continue. So, now then. Any antigen, so here's the red dot, this could be a protein. Okay? And this is the antigen then. Okay? And a typical antigenic protein often will have more than one epitope. Okay? Or more than one copy of the same epitope here. Okay? So any antigen that contains either more than one different epitope or many copies of the same epitope is a multivalent antigen. Most proteins that are foreign proteins, most antigens are going to be multivalent antigens. It's like an unruly child. I keep thinking if I ignore it, it'll just stop. But it becomes ever more insistent and irritating. Now, I tell you what, you can hear me pretty well without this. How about back rows? Are you hearing me pretty? Oh, I've turned this off. It's still. I'm going to try on amplifying. I hesitate to do that because I am kind of a soft-spoken guy. Somebody, please shoot me. Thank you.
course. To come back on because it's still cooling down. I have plugged some cables down there that hopefully can connect to the speakers.
Okay, so these won't have the top one going on,
Yeah.
but I won't be able to use the microphone.
The time's really not, especially if that's what's causing it. It's definitely the speaker. The speakers are really back now. Here we go.
While you're at it, you can reach one of your backup speakers. I know you always keep on.
Yes, I can actually go to the members. Yeah, no, yeah, I think we have one right there. So, go get it real quick. The projector should go
Let's try to continue on with this, all right? Page 34, page 34A. This is an important concept, and there'll be a question on the first exam, testing your understanding of this concept. Very simple question I'll ask you. It's a very simple concept. It's the difference between the term affinity of an antibody protein versus the avidity, A-V-I, avidity. Okay? The affinity is a measure of the tightness of binding of one of the arms, let's say, to its epitope on the antigen. So we have this complementary surface of the antibody, and here's the target epitope it's going to bind. Well, in real physical reality, when we say it binds, it doesn't come on and stay there unmoving into infinity in time. It comes on, after some period of time it will come off, some period of time it will come back on again. Okay? Now, for some proteins, it's a real strong, maybe a lot of plus minus electrostatic stuff here. It's going to come on, it's going to stay on for maybe a full second or even 10 seconds, where it gets some water molecules collide with it, knock it off, and then it can come back on again. Okay? So the time average over for the time, if 90% of the time it's still bound to the epitope, it has a high affinity. If it's coming on, coming off, coming on, coming off, it has a low affinity. Now, consider an IgM protein. An IgM protein, do you remember that it's a pentamer? And so it has 5 tails, each with 2 arms, and another guy with a tail and 2 arms. So 5 of those guys, their 2 arms, means we have 10 antigen binding sites, right? Each of the 5 guys has its own 2 hands. Okay? So an IgM has 10 binding sites all over the surface. When an IgM, a pentameric IgM, binds the surface of a bacterium, each little hypervariable tip of the V box of one of the 2 arms, it may not be the tightest, may not have the highest affinity. Maybe it's kind of on off, kind of on off, whereas a good high affinity selected IgG antibody arm is often on. 5 seconds later it comes off and it's coming back on again. High affinity. The V boxes of an IgM protein may have pretty low affinity. On, off, on, off. So, but at any given, but that IgM, some part of those 10 antibodies, half of those 10 antibody binding sites, antigen binding sites, are on at any given moment. they're going to be off again pretty soon, but because there are 10 of them, there's always somebody attached to the surface of the protein, okay? So that pentameric IgM is it's bound to the surface of that bacterium. All right? It may be losing it, but so the IgM has high avidity. Avidity is the amount of time that the whole antibody protein is staying attached overall. An IgG has 2 arms, so one could be coming on and off, the other one's coming on and off, but at any given moment, one of them is probably going to be on. So it has good avidity. IgM, pentameric IgM, has typically the highest avidity. Okay. In individual, for the pentameric IgM, each of the 2 arms may have pretty low affinity, but there's 10 of them. At any given time, as I keep saying, at least half of those 10 arms of IgM are binding to the surface of the bacterium at any given moment. So if I run that into the ground, you understand avidity versus affinity. Okay.
Can you repeat what avidity was?
Okay. Avidity is a measure of what fraction of the time the whole antibody protein, IgM or IgG, is actually attached to the surface of the bacterium, let's say. Affinity is a measure of how much an individual hand of an antibody is how much time it's spending on the epitope. So, if we had made a synthetic IgG and we kind of broke off one of the arms, and now we've just got one arm, it may have pretty good affinity, but when that one arm comes off, the protein, the antibody, can just diffuse away. Okay, with the bivalent IgG, one it may be a pretty high affinity, but one hand may come off, but the other hand is still attached. So the antibody protein is attached to the surface of the antigen. That's the avidity. Okay. I've seen many thumbs up. I get the idea that we can move on in this from this topic.
You're set up for now. I'm gonna go look for a clip-on. This is only here right now, but I'm gonna go look for the clip-on one. Yeah. It's gonna, might take a second.
Can you turn this thing off?
It's already on.
So I just have to increase the volume or something?
It's actually down here, because it's a physical speaker.
Testing, testing, testing. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5. 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3. I think a little bit over amplified. 2, 3, 4, 5, 6, 7, 8, 9. 1, 2, 3, 4. I think I can live with this, although I'd like to be a little bit more amplified.
Yeah. This is
That's what we got.
Yeah, this is what we got. And I'm gonna look for the other, I'll go
Okay.
look for the other one. I'll be back soon.
Okay, thanks. Okay, so again, avidity. The first antibody that's made after you get to protection is going to be IgM. It's rough and ready IgM. There's not been somatic hypermutation and selection to get a really high affinity. It often has pretty low affinity of its individual hands, but there are 10 of them on that pentameric IgM, and so it has very high avidity, so it's on the surface of the bacterium, and it's really excellent at activating complement. So we can get by with a low affinity IgM because of its high avidity, and that's useful for activating complement factor. Okay. I don't think I'm the least bit amplified, but I'll keep holding this. We now are going to just briefly go through a little bit more detail on epitopes. This is the most responsive touch. computer system here, it's got like one functional neuron. Okay. Okay. So epitopes, as I said, can be a string of adjacent amino acids on the surface of a protein, or these would be sort of called linear epitopes, or it could be what's called a conformational epitope, whereas only in the fully folded native protein do the amino acids are brought close enough to each other for the antibody to be able to bind them. So this antibody made against the native protein will work fine if you expose it to the native protein. If you immunize the animal with a folded native protein, it will make antibodies to a conformational epitope. If you then do like Western blots and you have SDS denatured the protein, it may have much of the protein may now be still in an unfolded form on the Western blotted cellulose paper, and the antibody won't see these two things adjacent to each other because of this denatured. And this picture here, I'm not going to waste too much time on it here, it just shows that, again, now here's a hypervariable loop which extends deep into a little channel deep into the protein. I'm sorry, here's the protein here with an extended epitope here that goes deep into a like a channel structure and formed by the hypervariable sequences here. Or we can have interactions like this, or a raw surface that's interacting and so forth. Now, I want to talk about monoclonal antibodies. I'm going to go into some detail on monoclonal antibodies, and there's going to be a question about monoclonal antibodies that involves the detail I'm about to give you in the next couple of pages of your okay, so everybody, slap cheeks and wake up over here because on the test. Now, forget about this. I don't want to hold this, but I can't put it down. Okay. So, if you get bit by a rattlesnake, okay, you might survive it, especially if you're a big burly guy, at least 320 or something like that. If you're a little child, you might die from the rattlesnake bite. And under any circumstances, it's going to be really painful. And it would be nice to get to a hospital where they could give you a shot, an anti-venom shot. It's called a shot. The shot that they're giving you is antibodies against all the various toxic proteins in the rattlesnake venom. So that comes from taking rattlesnakes. There are guys who make fun of doing this. They grab the rattlesnake. The rattlesnake is pissed off. And they put his fangs into a little rubber thing, and the venom gets squirted through the fangs, through this little rubber sheet here, and into a collection bottle here. And so they collect a couple of quarts of rattlesnake venom, okay? And they send that off to the factory. The factory takes that venom, inactivates it, okay, so that it's no longer toxic, and then injects it into sheep or cow, some large animal, okay? And that sheep or cow will make antibodies against the proteins in the rattlesnake venom, the inactivated proteins. Okay? And then you bleed the cow and collect the serum, the antiserum from the cow, and that serum, which is what you get when you collect blood and let the blood clot, the supernatant of the blood clot is the serum. Okay? So you collect, and if it's got antibodies to something of interest, you call that an antiserum. And then they sell that, make a nice profit, I'm sure, to emergency rooms and so forth, and you can get an injection. It would be nicer if we could make, and we now can do this, there's a whole technology and industry around this, monoclonal antibodies. So it's nice if you could just buy nice, clean, pure preparations of a particular antibody against one protein and another, two or three of the most important proteins. Right? So, maybe this is not the best example. For example, for many on you, I forgot to do, am I pregnant? Oh, God, no. Take a little test, okay, changes. Nothing, okay? Or, oh, am I happy or am I afraid? It's positive, okay? So that color change is based on an antibody recognizing a certain molecule in your body fluids that tells you if you're pregnant or not. So if you're dealing with crude antisera from pooled serum from a bunch of sheep and so forth, there can be variability from lot to lot. There can be some difficulties there. And again, having pure monoclonal antibodies against a particular antigen are much more reproducible, effective, they're really better all around. We also now use antibodies to treat cancer and many other diseases, purified antibodies directed against inflammatory proteins or cancer-related proteins and so forth. And again, monoclonal antibodies are key to all of this. They're pure, effective, very cheap at this point now. So, the way this works is, and this was work done by a guy who was actually a postdoc in the laboratory, but very famous immunologist. In almost all cases like this, the big famous guy in whose lab the postdoc was working, he gets the Nobel Prize. In this case, the postdoc and the big famous guy shared the Nobel Prize. So justice, we love to see it. Okay. So here's what the postdoc set up. He wondered if he could make monoclonal antibodies, and he had the idea to do it this way. He has a bunch of, he's immunized, in this case a mouse, with some foreign protein. Okay? Now, typically, when you interject a foreign protein into any animal, including a mouse, there'll be at least one B cell that makes an IgM on its surface that can react with an epitope on that foreign protein. But usually there are 2 or 3, maybe even 4 B cells that have complementary IgMs to that injected protein. So you'll get 3 or 4, maybe even 5 different B cells eventually becoming secreting plasma cells, secreting their own unique IgM against one epitope or another of the foreign protein that was injected. So here are the 3 B cells that became activated when the mouse was injected with a foreign antigen. They're each making a different IgM. Okay. Now, and he's going to pull these out. It turns out to be convenient to just take the spleen. The spleen is an organ filled with essentially little lymph node-like structures. So if you immunize the mouse and then take out the spleen, you've got lots and lots of activated B cells in the spleen. So these are these B cells that have been activated. Now, this mouse lives in a cage in a dirty old animal room and so forth, and he's got all kinds of B cells in him that are memory B cells or whatever from all the I think this morning he encountered some foreign thing.
I can't find enough on it, the other one, so can you make do with what you have?
I'm going to make you.
All right. And we'll get it right on this computer sheet though, so sorry about the inconvenience.
So, here's a bunch of B cells. Now, I said first a minute ago, these are the 3 B cells that happen to recognize some different on a single foreign protein. Let's make it simple now. This is the one B cell that is against the antigen. And these might be some other B cells directed against some other antigen that the mouse saw sometime during this life or along as part of the process. And this is the cell that we'd like to make immortal. Now you can purify these B cells and separate them from each other. So you could get pure yellow B cells here, and they would then be secreting antibody, but they only live for a little while. Okay? And part of that is that there's an intrinsic number of doublings that any cell can undergo. Okay? And it has to do with what we call certain repeated nucleotide sequences at the end of the chromosomes. I think most of you know what telomeres are. Okay? And I think maybe most of you also know that every time the cell replicates its chromosomal DNA, that DNA gets a little bit shorter because you can't fully replicate a linear DNA molecule. You lose some information at the end. It's just intrinsic in the nature of DNA replication machinery. So eukaryotic cells have solved the problem by this. Okay, well, we'll just have a bunch of junk DNA at the end of the DNA molecules, and it doesn't matter if that gets shorter over time because it's just junk DNA. But after 50 rounds of DNA replication, you burn through those telomere junk DNA sequences, and then when you start replicating the DNA, you're burning into coding sequence DNA. Actually, before you actually begin doing that, the cell has machinery that recognizes that the telomeres have become too short and initiates a stop growing signal, no more DNA replication signal. So all normal cells can proliferate for a while, but then they stop. They go into a senescent state and then they eventually die. So these B cells are intrinsically immortal. Okay, they do not live forever. They die, like all of us. They die. Now, here are some cancerous B cells. These are B cells taken from a patient who had leukemia. Sometime back in the past, they've become an established cell line now, and these cancerous B cells, they're called myellomas. OMA is a suffix that has to do with the fact that it's cancerous. And myeloma refers to lineage of these cells. Essentially, all white blood cell cancer is what this is, but it's a leukemia. It's a B cell leukemia here. So these myeloma cells, when you get this kind of myeloma leukemia, okay, you have an overproliferation of B cells, and those B cells have become plasma cells. They're actually secreting antibody. And one of the things that will kill you if you get a B cell lymphoma is that you get such a high level of antibody accumulating in your blood because you've got 100,000 times more of these B cells than you should have, and they're all secreting industrial levels of antibody into your blood, so your blood is like viscous with all the antibody proteins in it and so forth, and that clogs up your kidneys and it's curtains. So, part of the process whereby a cell goes from normal, all normal cells are mortal, the process whereby any cell can become cancerous, it has to do with turning on the machinery to maintain the lengths of the telomeres. So cancer cells, every time they replicate their DNA, their telomeres are not getting shorter and shorter because they have telomerase to kind of remake, to re-add on the random nucleotide, if they randomly repeated junk DNA nucleotide sequence. So a defining feature of all cancer cells, including these myeloma, is that they can maintain the lengths of their telomeres, and therefore they can proliferate indefinitely. We say that they are immortal. So our postdoc says, here's the cell that I want that makes the antibody I want, but it doesn't last very long in culture. And here's a cell that makes an antibody that you don't want. It's useless. It's some endogenous antibody, whatever. But it lives forever. So let's take these cells, and step number one is let's look for a mutant that's lost the ability to make its own endogenous antibody. So we're going to take these myeloma cells and select for a mutant variant that no longer makes its own intrinsic antibody. And then secondly, we're going to select for another mutation in this linkage that we're creating, where they've lost an enzyme involved in DNA nucleotide biosynthesis. They are, we say, HGPRT minus. In a moment, we'll talk about HGPRT. So these cells have a defect in the ability to now mutated in the sense that they can no longer synthesize the nucleotides they need to do DNA synthesis. So they've got to be grown in a medium where we supply the missing nucleotide precursors. Okay. So these cells are wild type HGPRT plus. Okay. These cells are HGPRT minus. Okay. We've mixed the population of all kinds of B cells from the spleen of the immunized animal, take those B cells, and we mix them with an equal number of these myeloma cells. And then we add a molecule to induce cell fusion, polyethylene glycol, which coats the surface of the cells. Normally when one cell encounters the second cell, it sort of bounces off. But if the surface of these cells, the plasma membrane, is coated with this polymer, polyethylene glycol, the reasons are a little bit obscure. When the two membranes kiss each other, they fuse, okay? It becomes a friction. And they become now a hybrid cell, okay? So here we have this B cell fused with one of these myeloma cells, and here is now a fused B cell myeloma cell. The fused cell we call a hybridoma. Here's a fused yellow B cell with a myeloma cell. Here's a fused green B cell with a myeloma cell. So these fused cells, these are called hybridomas. They're called omas, hybridomas, because they now have the property of being immortal, just like the parent myeloma cancer cell. So the enzyme that was turned on in these cells that maintains the telomeres, that makes them immortal, that enzyme is now present in a hybrid cell. So this hybrid cells will now grow indefinitely because they've inherited the telomerase enzyme from the cancer parent. Everybody with me on this so far? And now also, these cells are secreting antibody because they have the antibody gene of the B cell partner. So now we have a hybridoma cell which is secreting whatever antibody the B cell partner had, and is now able to grow indefinitely because of the myeloma partner. Now there are going to be some, so we're going to have hybrid cells. We're going to have some unfused B cells. This is a spleen B cell here. So some of these spleen B cells will not have fused. And we're going to have some unfused myeloma cells. So unfused B cells, fused B cells, and unfused myeloma cells. Now we're going to put this picture of these three different kinds of cell types: unfused B, unfused myeloma, and hybridoma. We're going to put them in selective medium called HAT medium. And then in HAT medium, the unfused B cells, they're going to die. And also in HAT medium, the unfused myeloma cells are going to die. Okay? But the cells that will live are the hybridoma cells. They live in HAT medium. So a HAT medium selects out unfused B cells, unfused myeloma cells, and only those survive. That old Rhythm Blues song used to go. Now we have just the hybridomas. Now we've got to go through a series of dilutions and picking in cells, individual cells. We've got to dilute out things so that we select out a blue hybridoma cell from the other ones in one well of a multi-well plate that goes repetitively here. So you can then isolate pure blue hybridomas, pure yellow hybridomas, pure green hybridomas, and they're going to these individual. Sorry, not they're going to be individual wells of a multi-hybrid plate. And now we're going to test the culture medium of these thousands of individual wells and look for the wells which are producing an antibody to our antigen of interest, the antigen that we use to immunize the mouse. And then we find the one well of hundreds or whatever, that's making in which this hybridoma is secreting the antibodies. This is now a hybridoma cell. We can isolate it, grow it up, it'll grow indefinitely. You get large numbers in, you get pounds of these cells, and they're producing, tons of pure monoclonal antibody in their supernators. And now you can use that pure monoclonal antibody for medical purposes or whatever. So now let's go into just a little bit more detail of what I mean about HGPRT minus and plus and what HAT medium is. HAT is hypoxanthine, aminopterin, and thymidine, H-A-T. Okay. Aminopterin is a poison. Okay? It blocks DNA synthesis. But you can get past the aminopterin block by providing precursors that bypass the block that the aminopterin poison produce. Okay? So hypoxanthine is one salvage pathway, and thymidine is another salvage pathway. So here's the deal. Aminopterin blocks the conversion of the precursor molecule to thymidylate. Okay, aminopterin blocks this pathway in the biosynthetic pathway. Okay. Well, so if we provide thymidine, we just bypass this, and so even though the cell itself can't make thymidine, we can't make thymidylate, we can provide thymidine, which is thymidylate minus the phosphate. So thymidine comes into the cell will put a phosphate on thymidine, making it thymidylate. You don't need to know this detail of biochemistry. Okay? So aminopterin blocks the synthesis of the particular pyrimidine molecules here in thymidine here, and we can bypass that by just providing thymidine in the HAT medium. Okay? It also blocks the biosynthesis of purines here. Okay? There's a precursor that be converted to another precursor, a purine precursor called inosinate here, inosonate, and then inosonate can be used to make GMP and AMP. Of course, GMP goes to GTP, AMP goes to ADP. These are the deoxynucleotides here. So in the presence of aminopterin, we cannot make 3 of the 4 nucleotides that you need to replicate DNA. Okay? But we can then provide thymidine here in the medium, and then we can provide a molecule called hypoxanthine, and the cells have a salvage pathway. If there's a problem with this pathway working, all cells have a pathway involved that uses the enzyme HGPRT. Okay? And HGPRT can take hypoxanthine, and it can convert hypoxanthine to inositol, okay, IM3, thereby bypassing the aminopterin block. Okay? So, however, the B cells, they have the HGPRT enzyme, so B cells can grow in HAT medium, they can bypass the block here. But the myeloma cells, the unfused myeloma cells, we selected them to be HGPRT minus, so they can't take advantage of the hypoxanthine that's provided in HAT medium, and so they die. Is that clear? So HAT medium selects for the hybridoma cells, because the hybridoma cells, they have the HGPRT. The B cell partner has the HGPRT so that the B cell and the myeloma cell, now they're one single cell. The B cell is giving us the HGPRT enzyme, okay? And the myeloma cell is giving us the telomerase to now make us be immortal. Okay? So all that's written out down here. Now, one of the questions I'm going to ask you on the first exam related to this material, not the only question related to this material, but one question is going to be, in what way have these myeloma cells been treated, so to speak? How have we modified them to use them for the production of monoclonal antibodies? Well, one thing everybody remembers when I ask this question is they've been selected to be HGPRT minus, and therefore unfused myeloma cells, they die in HAP medium because they don't have the enzyme that would permit them to live in HAP medium, right? But the second thing I want you to remember, okay, is, as I told you, and is again down here, but at the bottom of the page, put a big, mark this, put arrows on it, okay? This is reason number 2, okay? Okay? They have been selected for the inability to synthesize their own endogenously encoded antibodies. Remember, myeloma cells are B cells gone bad, okay? They broke bad, okay? And they can't make, they've lost their own endogenous antibody gene. So their only job here is to provide telomerase to the hybrid hybridoma, so that it will now be immortal and be able to grow again. Now, I'd love to take a break, but altruistically, I think we should continue on. If some of you are at the point where you just got to take a bathroom break, you'll have to self-identify. I'll just let you quietly leave, but I'd like to press on here, okay, and try to get a better sense that I'm going to finish the material that I have. When I was in college, I did a junior abroad program. I was at the University of California. They had a program, I was in their German program in Göttingen, famous old mathematics and physics, lots of quantum mechanical stuff, and mathematics was developed in Göttingen. Beautiful town. One of the courses I took was a history of World War II, given by a guy who was on the general staff of Hitler's army. Okay? He was taking notes. He was the official historian, so he was taking notes as they were making all their evil plans, okay? Including the invasion of the misbegotten invasion of Russia, betraying his Stalin partner. That was the end of it. I mean, that was, it was God who put that misbegotten plan in Hitler's mind, because that was the end of the Nazis, okay? Their army froze to death in December on the steps of outside of minus 60 degrees centigrade temperatures. So he was there, but this was the evening class, and it was 3 hours long, no breaks. And I'm still at the point where I'm not just listening to it. I'm getting pretty fluent in German, but I'm still translating, you know. And by the end of the first hour, my neurons have lost all their ATP. I'm catching about every second word and so on. So anyway, I have some sympathy. I tell you, I'm going to go on for the second hour straight here. So I told you before about the mystery that the immunologists had. We know that animals can make antibody proteins with all millions of different kinds of specificities, and yet there's not enough room inside the nucleus of a cell to have, for every B cell to have all possible genes for all possible antibodies. It's just too much. Okay? So they thought, well, maybe there's a gene for some kind of weird prototype protein that can itself sort of induced fit kind of thing. Well, that was nuts too. And the answer to the problem turned out to be, as I said, all cells, sperm and egg, have in them little pieces of a gene that can be assembled to make a gene for an antibody protein. Okay? And in just in the B cell lineage, in the bone marrow, we go from what's called the germline arrangement of DNA with little boxes and pieces that are going to be assembled. In B cells in the bone marrow, they begin a process where they start cutting, throwing away and joining DNA molecules so that we bring these little bits and pieces into contiguous right next to each other. Okay? So inside, in the germline arrangement, we have a bunch of boxes that encode a B box, okay? Okay. Maybe, I don't know. Let's see, this is the light chain. This is the chromosome that's going to make a gene for a light chain. Okay. Now there are two flavors of light chains, okay, chocolate and vanilla. Lambda and kappa. So this is the arrangement of segments to make a gene for the lambda light chain. It's on chromosome number 22. On a different chromosome, number 2, we have a bunch of boxes that can be assembled and brought together to make for a contiguous array of exons here to make a kappa light chain protein. And then on a third chromosome, number 4, now you don't need to remember the chromosome numbers here, but on chromosome 14 here, we have a bunch of boxes that can be brought together to form a heavy chain. Okay? So let's start with the heavy chain. Okay? Now we have these bunch of boxes here, and we're going to be picking one of the boxes at random and joining them together. Down here we have a simplified blue box here that just says CU. This means this is a segment of DNA that codes for the constant domains of an IgM. U is the M, Greek for M. Okay? So this is the IgM heavy chain segments. So here's the IgM heavy chain here. It's got 1, 2, 3, 4 constant domain here in the finished protein. And the DNA that codes for these 4 constant domains is in this blue box. Okay. And now we have then, for the heavy chain, we have the variable region here. Okay. Now the variable domain, the V box here, the variable domain, is what we're going to make by joining these boxes here. They're going to come together to make a so-called VDJ set here, and that's going to code for the V box of a heavy chain. So here the constant boxes are in the blue part of the DNA, and this box right here, the beta sheet structure that's going to be the variable domain of an antibody protein is going to be assembled at the level of DNA. We don't have any gene to make this yet, and we're going to make the gene by cutting and joining. We're going to first select at random one of these 27 D boxes, and then pick at random one of these 8 J boxes. We're going to bring together to form a DJ pair. It could be D17 and J4 in one B cell lineage. In a different B cell lineage, it'll be D7 and J1 and so forth. Having made a DJ pair, we're then going to hold on to that, and now we're going to loop out the intervening DNA, and we're going to pick at random one of these 65 V, these red V boxes here, at random. So we might random pick V box number 3, okay? And then join V box number 3, cut out the intervening DNA, and then ligate the end of this DNA molecule here to the DJ molecule that we have made. So now we make a VDJ contiguous DNA sequence here, and that's now going to be made into a messenger RNA that will then include we'll read these D boxes off here. We'll make an mRNA that has the VDJ and then all the constant, the DNA sequence that shows all the constant domains. Now, I'm going to show you pictures to make this clearer. So that's how we assemble a functioning gene in a particular B cell in the bone marrow from bits and pieces like this. A similar thing for each of the 2 light chains. What we're going to choose here for the light chains, for the lambda light chains, we've just got 2 J boxes. I beg your pardon. Looks like 4 J boxes, or J1, J2, J3, I don't know, whatever. 4 J boxes here, okay? We'll pick one of these and we'll join it to one of the V boxes. So for the light chain, we pick a J at random and a D at random. There's 30 choices for a B. Okay. And then we have some different options here for the constant domain of the light chain. All this will be revealed as we look more detail in subsequent lectures. For the kappa light chains, we have 40 V boxes, 8 J segments, and just 1 constant. Any questions at this simple level here? I'm going to, if not quite clear, I'm going to be explaining more. So, here's how we then make this is at the level of DNA here. This is germ-like DNA. Every cell in our body, our liver cells, our neurons, they've got these boxes that are potentially could be assembled to make a gene for an antibody protein, but that never happens in the neuron or liver cell lineage. Only in cells which are going to be B cells do we activate this process here where we start cutting and joining here. Whereas, like I said, first we pick a D box at random and a J box at random. We have 6 choices for this and 27 choices for this. We pick one of these and join them together. Now we have the DJ pair, and then here's the stuff that codes. It's all we have pre-built in the nucleotide sequences that we need to code for the constant part of the protein. That's why all IgMs have the same amino acid sequence, because they all come from a single nucleotide sequence that all B cells have for the constant domains. Where a different B cell makes a different proteins because they make a different B domains. Here's how we make the B domains. So when I say we assemble the parts to make an antibody protein from boxes, I should say we assemble it. We have the parts to make an antibody, almost all there, except we've got to assemble the variable part, the gene that encodes the variable part of an antibody. And you do this by first making a DJ pair, and then having made the DJ pair, we're going to select one of the, whatever it is, 85 B boxes here. If this were E40, there'd be 40 more downstream for it. That's going to get looped up, thrown away, cut out and thrown away. Now we're going to grab this B box here and join it to the JJ. Now upstream from this, there'll be another 40 B boxes, but they're not going to be read into messenger RNA. There's a promoter right here in front of this B box, and we'll read this part into primary RNA transcript. But not yet. This is also at the level of DNA. So we have a DJ pair. Now we put a B box. We join that to the DJ pair. Now we have, in the DNA of a B cell, okay, we have a gene now that encodes a fully functional antibody protein. All the constant domains are IgM. They're right next to this process. So the first thing we make is we read through and we make an antibody protein with the IgM, C mu for IgM, constant domain tails. There are other constant domains downstream from this, and we can loop out and put them onto the assembled B boxes at the level of DNA. Thanks, again, getting ahead of myself. So it's at the level of cutting and joining DNA molecules that we then assemble a functional gene for a particular, to make an IgM protein. So we can do a little arithmetic here. Let's start with a heavy chain option. We can choose from any one. Individuals differ from each other a little bit here. In different individuals and in different populations on the surface of the Earth and globe and so forth. Some people have got 38 B boxes. Some people have as many as 46 B boxes. On average, we have, let's say, 43 B segments here that can be used to make a heavy chain of an IgM. Okay. And then we have 23 diversity boxes, and we have 6 joining boxes. Okay. So to assemble a particular gene encoding a particular IgM antibody. We can pick any one of 40 here, any one of 23 here, any one of 6 here. You can convince yourself that therefore the possible number of different heavy chain genes that we could assemble is going to be 40 times 23 times 6, which is 5,520 different IgM proteins, with different D boxes that you could possibly assemble just from the different choices on your menu. So that's pretty good diversity. That's so all this process we're talking about here now is the generation of diversity. So the generation of diversity can be in part explained just by the many choices we have in terms of joining B's and D's and J's together. Now notice that there are actually 9 different constant domain boxes that you could choose from here. These correspond to IgM, IgG, IgE, IgA, and so forth. That doesn't count towards diversity. Diversity is the variability in antigen binding. The tail, the constant tail part, has got nothing to do with diversity of antigen recognition. The diversity of antigen recognition is all about the D boxes here. So we ignore the constant domain options here, and the number is just these 3, B, D, and J. Now, there'll be a question on the test where I tell you we've got X numbers of this number. What is the possible diversity? I'm going to give you this number right here for the constant domain options. I'm telling you right now, don't get confused by the question on the exam. You're going to ignore, you're not going to add in this 9. You're not going to say 40 times 23 times 6 times 9. No, not 9. Okay? It's just these 3 numbers you multiply together to get the variability, the diversity of antibody proteins here that you can generate by this process. Okay? Same thing now then, to make a particular lambda chain gene, it's going to be an average of 30 V boxes times an average of 4 joining boxes. Okay, so it's going to be 30 times 4. It's 120 possible lambda-like chains that can be brought together in the same antibody. So to make an antibody protein, we've got to assemble a gene for the heavy chain, and then assemble a gene for a light chain. The light chain can be either kappa or lambda at random. So, possible of 120 different lambda light chains, and 35 times 5 is 175 possible kappa light chains. So the overall diversity and the antibody that you can produce, just based just on your choices from the existing menu here, is going to be this number times this number times this number, which is going to be 1.6 times 10 to the 6. So about 1.5 million possible different kinds of antibody specificities you can make just at the level of choosing from the menu. So that's pretty impressive, but actually the diversity is many orders of magnitude greater than 1.5 million. So where does the additional diversity? Well, this is the one question. So the question you're going to have on the exam is about this basic simple thing here, choosing from the menu. Now, but I want to tell you that there's much more diversity that comes into play. Let's see. First, a little bit more detail here, not on the test. I want to write this down. Not on test. I'll pause while you write that down. When I see for, let's say here, for a particular, let's say picking a particular J and a particular B for a light chain here. Upstream from one of the particular J's is this so-called recombination signal sequences here, RSS sequences. And they have a little sort of substructure to them. We have a heptamer, and a spacer, and a monomer. 7 nucleotides, spacer, and then non-nucleotides. And then downstream from all the V boxes you're going to choose is another RSS sequence. This is called recombination signal sequence 1. This is RSS sequence 2. These two have complementarity to each other, and they're going to line up. They're going to be recognized by a protein here called the RAG protein, shown in blue. RAG protein, RAG1 binds to this sequence, RAG2 binds to this sequence. Actually, I misspoke when I said these have complementarity to each other. They each bind a protein, a pair of proteins, and these proteins are sticky to each other. So when the RAG1 protein binds this, and the RAG2 protein binds this, then we'll form these RAG1 and RAG2 proteins bind to each other. That core extrudes out a kind of a loop like this, okay, because of the binding of the RAG proteins. And then that's going to recruit other enzymes that are going to cut the DNA here and here, and this is going to get thrown out. We're going to loop this. This piece is going to go away as a loop of DNA, and we're going to then ligate the end of this V box segment here to the beginning of the J box sequence. They're going to get ligated together, and here we've made the BJ joint. Now, and the same thing happens with capital chains and the DDJs and stuff. Now the deal here is that when you do this cutting and joining here, you introduce, in effect, kind of random nucleotides. And the random nucleotides that are put in are called P nucleotides and J nucleotides. So when we attach the V box to the J box, we don't just directly take the V box end and the J box end and ligate them together. We insert random nucleotides. And we might have 50 nucleotides that we insert. And now that so the VJ joint has got VJ and then a bunch of random nucleotides here. Okay? And those random nucleotides can be random number, but they fall into two broad categories, P and N. So let's take a look at that. Page 41A. Not on test. Not on test, but I'm just going to explain it to you anyway. Okay. Now, when you cut here, okay, you make a cut such that the Watson strand is going to be chemically attached to the Crick strand down here. So now we have a little Watson-Crick continuous thing going on here, and the same thing over here, okay? And then we're going to cut this little hairpin thing here, E, and here on this one. So we'll cut the sort of Crick strand down here. We cut the Watson strand up here, and this now opens out, and we look like this. Okay. The end of this right here used to be kind of around attached to this. Now we cut here, now it's straight out. Okay, and this used to be attached to here, and now we cut here, and now it's straight out. Okay, these they are Madam, I'm Adam palindrome nucleotide sequences here. So these are the P nucleotides that are in fact randomly inserted between the D and the J sequences here. This is going to get, now we're going to take this and we're going to start ending by a terminal deoxynucleotide transferase. Terminal deoxynucleotide transferase. Terminal deoxynucleotide transferase. Okay. That just starts adding random nucleotides onto this and to this. Okay? And since they're added randomly, there'll be some just by chance some little regions here where we'll have ATGC pairs here. Okay? And now cellular enzymes here will, this part here that's not convoluted, that it cuts away. this part here that was not counted, the second cuts away. So it's just 2 or 3 contiguous base pairs here. And then we fill in, we make this sequence, the rest of this complementary to this, and then we use this as a template to add this to this. So now we end up with double stranded Watson quick here, a randomly added nucleotide, okay? So these are the sort of the, what they're called, the non-germline coded here, the N nucleotides here. These are the non-nucleotide coded enzymes. I'm going to now I'm going to show some more detail on this. Again, this more detail, I'm not going to ask you this level of detail on the first exam. Okay, and again, this is just showing you, here we have the pyramid form here, watching the quick strands here. And it's going to get cut open. They now extend outwards here. We're going to add this is the palindromic nucleotides here, the 2 nucleotides. Then the TDT enzyme starts adding random nucleotides onto the end of this. Some of them finally will give us a little base pairing here. So we'll cut off the stuff that's not base paired and then fill in the template synthesis here. And these random, all these extra nucleotides, these are the non-coding nucleotides here, or N for non-germ-like nucleotides added here. And up to 20 nucleotides can be inserted there. Now, these P and N nucleotides, since they're random and they're random length, we have a diversity box here, which if read in sequence, in phase with the V boxes, are going to be within a reading frame here. If you add or delete nucleotides in the middle of a reading frame, you're out of the reading frame, okay? And you start coding for nonsense protein. Well, for antibodies, there's no such thing as nonsense protein. It's all grist for the mill, it's just diversity. But you can also, once you're out of the reading frame, you can find something you're arrive at a stop codon. Okay? You want to be stop codons. So when B cells, we're going to get into this in more detail later on. But in the B cells, in the bone marrow, they're doing this process of creating a functional immunoglobulin N variable region. A lot of these B cells, they do all the stuff, P and T nucleotides, all that happens, but then they made a gene that has a stop codon in the middle of it because of the insertion of the N and P nucleotides, and that's a sorry Charlie, you're dead. B cell, that's as far as he ever goes. So there's a lot of carnage in the process of making a successful B cell that has assembled a functional gene that can be read into a functional protein. Again, we'll come back to that. Now, I just want to show you one more thing here, which is, I think in your notes, you have page 41E in your notes, I hope. Oh dear, some of you have it, some of you don't. If some of you have 41E, raise your hand. Yeah? Where did you get your notes?
At the copy center.
At the copy center. I'm sorry I did not update the online stuff. This is one of these things where I recently put this in and forgot to put it into the online. Okay. I will try to remember. This is useful information, and I'll try to remember to pop that into the online things just for you. So let me just show you here a little bit more detail on this, so you're kind of clear on what's online. Okay. So we so these B boxes here, anyone at 50, for this, now this is a heavy change we're going to assemble here. We've got 50 V boxes. Each V box has the most of the variable chain, the variable domain part of a particular antibody, okay? It's got this the complementary the variable region, hypervariable region number 1 and hypervariable region number 2, okay? These are the loops that are the business end of it here. And what we're actually assembling in this process is the hypervariable region number 3, or the CDR3 loop. The different V boxes here, the 50 different V boxes, they make, basically they are pre-made hypervariable 1 and 2 loops, or CDR1 and CDR2 loops. That's built in. And this is over the course of natural selection, encountering pathogens over the course of vertebrate evolution, including most recently 10, 20,000 years ago when we finally formed villages in the Middle East and then domesticated animals and then encountered animal smallpox and all kinds of other nasties like that that produced the Europeans who invaded the Americas in 1492, sailed the ocean blue, that thing, okay? That's all been selected for, all right? So a lot of these V boxes, they're not just random. There's been some selection there. They've been useful V boxes for us to have as a species here. But just in case we then make all possible hypervariable loop number 3 here, okay? There was no HIV in human population. There's no HIV virus up until quite recently, okay? So we don't have V boxes to help us deal with HIV. We've got to generate a V3 loop to deal with HIV. Okay? And that's where here's the J box here. Here's the diversity box here, and here is the end of the V box here, and we're making the V3 loop right in here, right here. This is the diversity we're going to get. It's part of the diversity in the hypervariable stuff, and the hypervariable loop is just essentially a framework region that the J box is providing. But then the hypervariable loop here that makes that one big loop here. That's the joining of the V and the D and the J, and the inserting of random nucleotides. So we insert random nucleotides between the V and the D box, and then insert random nucleotides between the J and the J box. And that's where, so all the lot of important features that make us totally new specificity B cells with these approaches is the hypervariable loop number 3. That's where you, and that's, a lot of that is because of the inserting of the random N and P nucleotides. Now, so here's a little bit sort of zooming out again to get a little bit bigger picture here. We've been talking about how we assemble a particular V, a particular G, and a particular J. We join them together here. And then right adjacent to that now is the sequences that code for the constant domain parts of an IgM antibody protein. Well, when there's a promoter here in front of these V boxes, each of the V boxes has a promoter sequence in front of it here. So when we bring this particular V box number 17 to the D and J, that brings this promoter close to an enhancer sequence here, which is just in front of the IgM coding sequences here. And now this brings transcription. This permits us to assemble transcription factors on the promoter here. So so that we can now read through. So as soon as we have created this VDJ box, we've also activated transcription of messenger RNA off of this DNA sequence that we created. Okay? And the next page of your notes illustrates this. Again, each V box has its own promoter. When you bring a particular V box close to a DJ pair here, that means the enhancer that was always here, this is too far away to work on the V boxes here, but when we brought the V box close, now the enhancer can recruit transcription factors to the promoter here, and now we'll make primary RNA transcript off of this DNA gene that we created. Okay? Again, this level of detail is not on the test. You don't need to remember about how bringing the V-box together brings it close to the enhancer, which then will permit transcription. Don't worry about that detail. I'm just telling you. So now, in the very act of creating a functional V domain of an IgM antibody, this is the IgM, the constant domains for the U, Greek M, okay? This is what IgM. Okay? So we're going to make a primary RNA transcript. And the RNA is going to start making, we're going to copy RNA off of the DNA beginning here, and reading on down, and actually read on down past the IgM constant regions and into the IgD constant domain regions here. And so we make a primary RNA transcript that can be spliced in any one of two different ways to give us either mRNA for IgM or mRNA for IgD. Okay. So here is more detail here. Here's the DNA. We assembled the VDJ box. And we brought now the promoters activated. We make the primary RNA transcript. And that reads on through the DNA and it copies into RNA the C mu sequences and the C delta sequences, D delta for the IgD. Okay. So now we make a primary RNA transcript. Now we can splice that. We can then make a cut here, throw this away, and add poly A onto this. Now we have an mRNA that codes for an IgM heavy chain. Or we can splice it so that we cut here, splice donor site here, and then we use this splice acceptor site. So the light blue gets thrown away, and we join the dark blue onto the V box here. And now we have the IgD constant here. So from the gene that we assembled, we make a primary mRNA transcript so that we can go either way, okay? And about half of it, I think, I'm not quite sure the ratios, I think most of it tends to go to IgM, and a good portion of it, though, gets spliced so that we make IgD. So that on the surface of the B cell, that's going to be then released to the bone marrow into secretion, it's going to have a mixture of IgM, integral membrane protein, and IgD, integral membrane protein on its surface. I've been emphasizing all the time that it's IgM, but it's actually a mixture of IgM and IgD. Weird, mysterious IgD, it's there. And that's a RNA splicing event there. People get lost a little bit. Are you clear about this? You're going to need to be clear on what's happening at the level of DNA and splicing RNA. And don't get confused. When we use this term splicing, we're usually nowadays referring to RNA cutting and joining. Okay? When we cut and join the DNA, sometimes we use the term splicing, but it can get confusing. I'm going to say in our class we'll talk about DNA recombination events. Talk about the cutting and joining of DNA. And here is now there's a little bit more here now. So, we're focusing now on just IgM here. Forget about IgD for the moment. So we assembled at the level of DNA. We did the D and J and the BDJ. We assembled at the level of DNA here. DNA here. We assembled the V, and now the promoter is going to get activated. Now we're going to make a primary RNA transcript here. Okay. Now, not shown in this picture is that we actually read out through to immunoglobulin D constant regions downstream, remember, but just not focusing on the IgM here. We have now a messenger RNA here. This is the primary RNA transcript. This is a splicing step right here, where we're going to splice out intervening intronic RNA here. And now this is the messenger RNA where all the exons are contiguous. And we translate this from on the ribosomes to make this protein here. Okay. Now we have sequences down here that, depending on how we splice it, could include splicing in coding sequences for amino acids here that are going to make this be an integral membrane protein or be a secreted protein. So in this picture, we've spliced in the yellow transmembrane segment here. These are hydrophobic amino acids that are formed by alpha helix, a 20 amino acid hydrophobic amino acid. That's going to, a protein with that is going to be a transmembrane protein here. But now, once that B cell who's made the gene for an IgM, and it's expressing it with the transmembrane coding sequence here, so it's got surface IgM. When that B cell making surface IgM, if it were to get activated by encountering its cognate antigen, it's going to become a plasma cell, and the plasma cell is now going to go to secrete the protein that was made here. Okay. So this is again an alternate RNA splicing event here. So originally in the B cell here, we're using the membrane coding yellow exons here. And so we have these hydrophobic 20 amino acid alpha helical segments that are going to be the transmembrane part right here. But then if this B cell is going to become a plasma cell, we change the splicing machinery, and now the splicing machinery will splice out these yellow integral membrane coding exons and splice in these hydrophilic amino acid coding sequences here. And now this is a protein that will not become an integral membrane protein. This is a protein that will go by the secretion pathway as a soluble secreted protein here. So this change from a B cell making integral membrane IgM to a B cell become a plasma cell secreting, that's again an altered RNA splicing regulation. Okay, well, toughen your butts, we're almost there, we're getting there. So, all this time, I'm talking about IgM that has an integral membrane on the surface of B cells. Okay. At the same time, when the B cell completes its gene encoding this IgM, it will at the same time then start to activate the expression of some proteins that have immunoglobulin folds in them. They're called Ig alpha, the gene for protein called Ig alpha, and a gene for protein called Ig beta. Okay. These are also transmembrane proteins. Now, IgM has a tiny little bit of sequences in the cytosol, but there's like 2 amino acids or something like that. So it needs to recruit the Ig alpha beta partner proteins. They have longer cytoplasmic extensions here. And when the IgM binds the antigen, this is now going to function as essentially like a growth factor receptor, and it's going to initiate growth factor-like signal transduction pathways here. The actual signal transduction molecules that are involved are going to be recruited to cytosolic tails from the Ig alpha and Ig beta proteins here. Now, for test one, do not worry about this. We'll come back to all this in more detail in a subsequent lecture and subsequent tests. Okay? I'm just telling you now that there's alpha and beta that come into play, and they turn this protein into essentially a growth factor receptor. The growth factor is the antigen, basically. Now then, I've been telling you all this time that every B cell precursor that's starting off in the bone marrow to become a B cell, okay, it's going to go through the DNA recombinant cutting and joining events here to produce an actual functional coding sequence for an immunoglobulin hypervariable region. So that B cell has generated one of the million possible ways you could, by random nucleotide insertion and so forth, and make a gene for a protein that's going to be an integral membrane protein. It's going to stand here like this, and it's going to have hands like this that are the antigen binding regions here. And every B cell produces 10,000 of these guys all over its surface. They all have the same little hands. Okay? The next B cell has different hands on it. Okay? Now, that B cell has produced, just by chance, an antibody molecule that will have some kind of decent affinity for some epitope on a foreign antigen that's come into the body. Okay? And so that B cell will bind to that epitope, whatever kind of affinity, okay? And then that one B cell will start to proliferate with the help of T helper cells, remember, and differentiate into a plasma cell. Well, so you start secreting first IgM, kind of rough and ready, whatever you had made by your DNA rearrangements here. It ain't the greatest, but it's pretty good. Okay. But then those first B cells that are proliferating because they encounter the antigen, some of them go right away to plasma cells secreting IgM. The rest of them, remember, go to the B cell area of the lymph node where they proliferate. And during that proliferation process, they're replicating their DNA with all perfect fidelity for all the other thousands of genes and so forth. But when it gets to the hypervariable regions here, mutations are going to occur. Okay? Now, it's actually portrayed as if DNA polymerase makes mistakes. Actually, DNA polymerase works as usual, it's beautiful, but there's another enzyme that will come along and modify the nucleotides and therefore change an enzyme called the cytidine deaminase. Okay, activation-induced cytidine deaminase. It converts cytidines in the DNA to uridines. Okay, well, completely. Most of the time, that's going to maybe produce stop codons or make a really worse functioning protein. But every once in a while, this mutational agent creates better gene that makes for a better protein here. And this enzyme here acts just in these regions right here, the CD, the complementary, the hypervariable groups here. Here's the region in the DNA here, a bunch of different B cells here, with 1 week after immunization, you see mutations occurring in some of the cells in the CDR2 loop, and more of the cells in the CDR3 loop at the light chain of the heavy chain, and the same thing with the light chain. And now 2 weeks later, we see more mutations occurring, not just in CDR1, but also CDR2 with CDR1. They're not all exclusively in the hypervariable part here, but they're kind of clustered right around close to it here. And so these are the AID enzyme-produced mutations in the DNA in individual cells. And so all this is called somatic hypermutation. Somatic hypermutation. So we had a cell that was making a perfectly good antibody, not perfect, but pretty good, okay? It now goes into the B cell, and it proliferates, and while proliferation is going on, we have all this extensive somatic hypermutation, And most of the B cells are much worse off at the end of that, but a few of them are way better off, okay? And now all this collection of clonal descendants here, some of them better, some of them worse, some of them pretty much the same, they all are going to compete for the antigen, okay? And it's devil take the hindmost, okay? When I was a little boy, the ice cream truck would come by. I was at Inglesboro all my parents lived here. And the ice cream truck would come by, and the little English children would all get in line, and they would wait their turn in line, and it was all orderly. And then later on, as an adult, I was living in Italy for almost a year, and the Italian children, well, the B cells at this point, they're like the Italian children. They are just devil take the hindmost here. And somebody may get a hold of it, and someone else rips his hand off and he grabs it and so forth, okay? And the winner is the one who's got the tightest binding, okay? He's got the ice cream and he ain't letting go, okay? And so he gets the goodies from other cells that are supporting him, and that is very Darwinian. He survives, the rest don't. And you select then, among the population of hypermutated hypersomatic you select for this, the mutant B cell that's making an even much higher affinity. So here we have then a summary of what I just been telling you about the so-called generation of diversity. Immunologists, professional immunologists, who are by and large atheistic, not God-fearing people, okay? They're secular humanists, and they regard any believers with use tolerance, okay, for foolish children who know nothing about reality. I say this as a Christian myself, okay? So, but anyway, these are not God-fearing people, so I used to like to make a joke about how does diversity occur, okay? Well, they say sticker, sticker. God does it, okay? Well, they have noticed that generation of diversity. Now, the mechanisms by which God acts are, as we've described. Okay, I'm not going to repeat it, but there it is again. It's kind of summarizing what we've been saying so far. So, now, the one thing we didn't get to quite yet is all of this we talked about here is that the generation of diversity is the VDJ part of the protein, CR3 loop, hypervariable 3 loop. Okay? But we also have, once we've made a functional variable part of the antibody, that's the business end. Now we can join that to any one of a number of just different constant regions, different tails and so forth. First off, we make an IgM tail part here. But later on, after somatic hypermutation, after we fine-tune it, now we can then change its tail. And different tails are useful in different ways. So, I'm going to talk about that. This is the last part down here. Immunoglobulin constant region DNA rearrangements that switch the heavy chain with constant isotype. So let me remind you that we have these different kinds of immunoglobulins. Okay? IgM, D, A. We talked about this earlier. This is also on page 33E of your lecture notes. This stuff here is the same as what's on page 33E. You should know this for the test. You should know why we make IgM or IgG or IgA or IgE and the properties of all these things. So, because of what's called class switching, which occurs after the somatic hypermutation, we produce antibodies with the same antigen recognition specificity, but with different tables, okay? So there's IgM or E or G or A. Okay? Don't worry about memorizing this table or anything like that, okay? Just know, as we said, IgM made earliest, it's secreted as a pentamer. IgD, God knows what IG does. IgG, actually there's 4 different subtypes of IgG, maybe IgG1, 2, 3. I just want you to know that IgGs in general are most abundant in the blood. Okay, here's serum level here. IgA, actually there's 2, so there's IgA1, IgA2, don't worry about subtypes here. IgA is secreted to the gut, into your tears, and so forth. And the IgGE, which is in allergies. Again, don't worry about this table here, but IgM, it's pentamer, I keep telling you, pentamer. How many binding sites on IgM? 10. Okay, it's the best at activating complement. IgG is really good at opsonizing. Remember, opsonizing is you coat the surface of a bacteria with something like antibodies, that we say that bacteria has been opsonized. It makes it really tasty for the macrophages to eat. Okay, just like nice seasoning. Coming good. It smells so good coming in. Okay. IgA is secreted, so that even though IgG is the most abundant antibody in the blood, we make 5 times more IgA. It's just that IgA is not in the blood. It is secreted into your gut and the lining of your lungs and so forth, all those internal surfaces. And the IgE is fighting off parasites and worms and things. When we secrete IgM, we turn on the gene for a so-called joining chain, J chain here. Here's J chain. And we have a J chain that then ligates together, comes together, and we then produce the pentameric form. 5 antibodies, 10 antigen binding sites. 1, 2, 3, 4. J chain is also used to join the IgA together to form a dimer. Okay. There's more information here that I'm not going to have time to talk about, but just remember that IgA can dimerize. Now, not all IgA, the IgA that's going to be secreted is dimerized, but the monomeric IgA is also has a function here. But don't worry about all that. Just remember, IgA can form dimers. And most IgA is dimeric, and most of it is secreted. Page 54A, new material in this last year or so. Page 54A, do you have that in your notes? Nope. I suppose you have it. $1 well spent. Anyway, not on the test, not on the test. And also, none of you else have this 54B, I didn't get this on. Not on the test anyway. Now, the last thing, then we'll take a break, and we'll make it quick, and we'll come back and push the buttons for our review. This material, graduate students, look up, pay attention here. For you, this will be on the test. Undergraduates, congratulate yourself on being undergraduates. Don't worry about this. But this, from your book, talks about how we've been able to use what we know about this cutting and joining of the DNA, starting from the germline arrangement of segments to the B cell version of it. Okay? And we have now used this in the clinic to measure, for example, how well chemotherapy is doing at eliminating, let's say, leukemia. So here, let's go into this. Undergraduates, I mean, you may be interested in this anyway, but since it's not going to be on the test, if you want to go to your bathroom break right now, I won't hold it against you. Okay, graduate students. Well, first of all, probes, radioactive nucleotide probes, are now commercially available. There is a probe now, for example, that will bind to a nucleotide sequence which is found in thicker V boxes. Maybe all V boxes, I don't know. So we have probes that will recognize a nucleotide sequence in the V boxes here. There's another probe, another radioactive nucleotide, that will recognize a sequence here in one of the C boxes here. Now, there's a restriction enzyme that will make in the germline, this is now the germline DNA, all cells have this, including granulocytes. This isn't just a cell type picked at random. This could have been fibroblast DNA or whatever, liver cell DNA, but they're calling it granulocyte DNA. In the granulocytes, we just have the germline arrangement of all these DNA sequences here. And a particular restriction factor will recognize a site here in the V box, right about here somewhere, and then a second site right about here somewhere, and then a third site right about here somewhere. So when we cut, if we treat germline DNA with this restriction enzyme, we will produce fragments. One fragment is this size right here, between this cut and this cut, we produce a DNA fragment this size. It will have the probe binding site on it. And this other fragment between cut number 2 and cut number 3, this is a bigger fragment here. It includes the D and the J and some of the C, and the C has the C region probe on it. Okay? So, if we then separate germline DNA on an agarose gel, okay, and these pieces, this piece being small, will run deeper into the gel, as it turns out. This piece being bigger will run less deeply into the gel. If we probe this gel with the C region probe, the C region probe here will bind to this smaller piece, the bigger piece here, this fragment number 1 is what lights up. And then in germline DNA cells, the probe that recognizes the V box here will bind to this smaller piece, which runs deeper in. So non-B cells will see these two bands lighting up with our radioactive probes. Now, if the patient has a B cell leukemia, then we'll have joined some V box to DJs and so forth. We will have produced in the B cell, since we have a B cell leukemia, those leukemia cells are started as finished B cells that became growth dysregulated and became leukemia B cells, but they are B cells, and they've done the rearrangements here. So in the rearrangement process here, we now no longer have this site right here. That's been spliced out, that's gone. Okay? And all that's left is this original site here, and the B cell version of it. Okay? And then this site right here. So we make a single cut. The restriction nuclease will now produce a single piece of DNA when it cuts here and here, okay? And that's piece number 3, which runs this far into the medium size, and runs this far into the ovules, and it will react with either the C region probe or the D region probe. Both of these probes will bind to this one piece of DNA here. Okay? Now, if you just take white blood cells from a patient, just take a bleed, throw away the red blood cells, get the white blood cells and break open them, get the DNA, run that out of gel. Well, each individual B cell will have sort of different B boxes and so forth on it, whatever. But what you wouldn't see really, because there's billions of different B cells in a normal patient. There's no one particular band that lights up there. There's millions of bands that are present there and so forth. But because in leukemia, one B cell, the clonal lineage, is like half of the B cells in the blood and so forth. You see the DNA in this one B cell like this. So as long as he's got lots of B cells in his blood, you see this thing here. As the treatment progresses, this band gets weaker and weaker and weaker and then disappears. It's a measure of the effectiveness of the chemotherapy. Okay, so, graduate students. You can read this, you can write it. I'm going to go take a break. We'll all come back here and we'll start our exam review. Those of you who didn't happen to bring that lecture 1 handout that I gave you all at the beginning, there are a few extra ones. No. I said that if you didn't bring lecture handout 1, there are